Okay, as we go through these different compartments, an important question arises: where are proteins in the cell? And cell biologists have two important tools that we use. So one are antibodies to localize proteins, and we use immunofluorescence microscopy to look at where they are. How many people have done some immunofluorescence microscopy? Maybe in another lab, and maybe in a bio lab or something, or maybe in a research lab, and look through the microscope at where the proteins are. You get really beautiful results. And then the other way that we use are tagging proteins that, with GFP, and we do that by using genetically engineering GFP in frame with the protein, and then using fluorescence microscopy. In this case, it's not immuno, it should just be fluorescence microscopy to look at the GFP tag proteins. And how many people have done that in a lab? Yeah, it's fewer steps and less can go wrong than using immunofluorescence microscopy. So more people these days in this kind of class have looked at GFP proteins. Some people might think, well, GFP proteins, why would you ever use immunofluorescence microscopy when you can just GFP tag the proteins? And both approaches have pluses and minuses. So let's look at those experimental approaches. So let's first talk about immunofluorescence microscopy. In that case, we have some kind of antigen. So this might be the protein of interest that you want to look at. And then we've talked about how you can have a primary antibody directed against any antigen. So suppose this is the LDL receptor. So the antigen might be the portions of the LDL receptor. We've raised an antibody, a rabbit antibody against the LDL receptor. And then we fix the cell, incubate it with that primary antibody, and then expose it to a secondary antibody, which might be, if this is a rabbit antibody, it might be goat anti-rabbit, and that secondary antibody is tagged with a fluorescence tag, and it will bind to the primary antibody, and then we look through the microscope at this right here, and we can see where the secondary antibody is, because it's fluorescently tagged, telling us where the primary antibody is, the rabbit anti-LDL receptor, telling us where the LDL receptor is. So if we were to do this with cells, we would see the plasma membrane largely glowing up. There might be a little bit of ER, because remember the LDL receptor is traveling there, but you end up usually not seeing much of the secretory pathway. You usually end up with these proteins, as it is where the vast majority is, which is on the plasma membrane. And just as we talked about with immunoprecipitation, you might be wondering, why don't we just tag the primary antibody? Why go through this secondary thing? And there's a couple reasons why. One is these purified secondary antibodies that are labeled really bright, are cheap and easy to buy. You could, $100 gets you a vial that lasts a year, or 6 months, depending on how much you're doing the experiment. They're cheap, and you don't end up screwing up your primary antibody by trying to label it and mess up the chemical reaction. So they're cheap and easy. And the second reason is that you get an amplification of the signal, because if you were to label this one, you'd only have one signal, fluorescent signal here. Look, in the secondary antibody can bind multiple sites, so we've amplified our signal here in this example fourfold by having 4 fluorescent markers. So advantages, it's easy, it can be applied to many separate cells, good sensitivity. Disadvantage, cells have to be fixed and permeabilized, poked holes in to get this antibody into it. If this were an internal structure, the cells are dead and you can't observe movements. But it's easy, it's not, you don't have to clone anything like you would with the GFP. If you're tagging the GFP, you need the gene. You can screen tons of cells with it. Any questions about immunofluorescence microscopy? Okay, so on the other hand, we have tagging the proteins with the green fluorescent protein, and in that case, we need the gene for the protein of interest, like the LDL receptor. We just do pretty simple genetic manipulations. You put it in frame with the gene for GFP, and then transfect that into the DNA into the cell. The cell makes mRNA, of course, and then the protein ends up being tagged with GFP. And then you look with immunofluorescence microscopy, this is still living, so you can see where the protein is going. You can look at dynamic movements. This was a huge revolution in cell biology. But if there, and let's see, I don't have disadvantage. The disadvantages of this are that there's a fairly big one, and that's that it's possible that putting GFP in frame with your protein of interest will alter its distribution or affect the folding of the protein of interest. So there are a lot of controls you want to do to make sure that you're really looking at the proper protein. The other thing is, in general, you're overexpressing this protein. It could be you're changing expression levels, and that could alter the results. But you do the proper controls to convince yourself that you're looking at the right thing, and this has been a super powerful. It can't be used in every protein because sometimes you kill the protein by putting GFP there, but when it works, it's great. Not only that, but since GFP has been isolated, there are variants of GFP from blue fluorescent protein to red fluorescent protein. So you have lots of different colors. You can transfect the oxygen into this really beautiful cell that has lots of protein moving around. Okay, questions about immunofluorescence microscopy or GFP and how we work. These have been used a lot in membrane graphics. Okay, so we're going to talk now about coated vesicles, but before we do that, okay, come back to reality. Hopefully that little bit of smile will clear your mind and you can start the wheels turning again. We're going to end now, in the last 20 minutes, we're going to start talking about the vesicles that move proteins from one compartment to another compartment. And the concept of vesicles moving from compartment to compartment is true for many steps along the secretory pathway, whether we're talking about going from the ER to the Golgi, or going from the Golgi to the plasma membrane, or going from the trans-Golgi network to the lysosome. So as we talk about, we were very specific about translation into the ER. Now as we talk about vesicle transport, I'm going to talk about just the best studied example of each of many of these, and we won't talk about each of those separately because there are things in common. This will make sense. So what's in common for vesicles? So when we're going from a donor compartment to a target compartment, so this might be the ER going up to the cis Golgi, or this could be from the trans Golgi up to the plasma membrane, or to an endosome or a lysosome. We have a vesicle that needs to bud off from the compartment. So this is a small, about 100 nanometer size compartment. So the bilayer needs to bud off, capturing any proteins that are soluble in the lumen. And then when that bilayer of the vesicle fuses with the next compartment, it will deliver the contents of that vesicle to there. And this diagram nicely shows what I was trying to say in words before. Look at these red balls. They are in the lumen of the donor compartment, like the ER, and they stay always in the lumen of that vesicle, and then they're going to be in the lumen, they're always going to be in the lumen until they get to the outside of the cell. If we look at this green stick figure right here, which is an integral membrane protein, you can see it was in the lumen of the donor compartment, so it's always going to be in stuck in a membrane, whether it's in the membrane of the vesicle or the target compartment. And then the portion that is in the, that was put in the lumen of the ER is going to end up being in the lumen of the vesicle, and then in the lumen of, and when this fuses, you have to imagine that. You can see, as here it's fusing right here. You can see that that remains in the lumen, and what was on the outside cytosol part is always in the cytosol. So if you think about it then, and look at this picture, you can imagine the energetically unfavorable event of taking this bilayer, which has this hydrophobic core, and fusing it into a vesicle, budding off a vesicle. So somehow these two bilayers have to fuse together. to make a vesicle, and then when that vesicle needs to right here fuse with the target membrane, and somehow these polar head groups need to intertwine with these polar head groups, and the hydrophobic core needs to meld with that. So there's cellular machinery that help aid that energetically unfavorable process. And there's another thing that the cell needs to face too. So how do we get the right proteins into those vesicles? And then how do we deliver them to the appropriate compartment? So this vesicle that's budded from the ER needs to fuse with the cis Golgi and ignore any mitochondrial membranes it faces, ignore the nuclear membrane. Remember that the ER is all over the cell, ignore the plasma membrane. So we have to get the right vesicle to the right compartment with the right stuff inside. So what are the regulatory proteins that determine that? And what are the proteins that make this vesicle able to bud off and able to fuse? And that's the story that we're going to cover in the next couple of lectures. And we're going to start with the coated vesicle, the coat proteins that fashion that vesicle and allow it to bud off from the membrane. And in some cases, those coat proteins also help select what's inside the compartment. So let's look at that. So vesicle budding is accomplished by coats that surround the protein. The coat protein, there are many different kinds of coat proteins in cells, or several different kinds. They all are peripheral membrane proteins, basically, that function as a mechanical device to bend and curve the membrane, a mechanism for selecting the vesicle, and we'll see how a small GTPase is used to regulate the process. As we get this vesicle right here, budding off right here, it'll be the coat proteins that help fashion that vesicle. And the coat protein, there again are many different kinds of coats. This has 3, but there's sort of 4 because clathrin can be coated into 2 different kinds. We'll see that there are 2 different, two vesicles shown in red here are the coat proteins that are responsible for budding from the ER and going up to the cis Golgi. We'll see that COP1 vesicles go back from the Golgi back to the ER. And then there are 2 different kinds of clathrin-coated vesicles, one that forms on the trans-Golgi network, and another one that's responsible for bringing proteins to late endosomes that will end up in lysosomes. And there's a different kind of clathrin coat that's on the plasma membrane that's responsible for endocytosis. So we're going to talk about these coats in depth. How many people remember clathrin from an earlier class? Nobody? One person. It might sound a little bit familiar. Clathrin's the protein that I worked on for some 20 or 30 years in using a microorganism, so it's my favorite coat protein. And then it was the first coat protein discovered. And then subsequently COP1 and COP2 coat proteins were discovered. How many people, does that sound sort of familiar, COP1 and COP2? I'm going to tell Dr. Buzzkirk and all the others, because I'm sure she covered it in some way. But it'll come back to you, because we're going to spend a lot of time talking about COP1, COP2, and the clathrin coat. So even if you don't remember, it's in there somewhere. We'll reactivate those neurons. Okay.